you can solve a lot of problems with plant biotechnology. For example, you can make a crop less prone to be attacked by certain insects. Yeah, so you get a better harvest. You can make plants adapt to salty soil environments, so you can grow them in places where you otherwise couldn't grow anything.
Welcome to the Ahmed Lab podcast, where we discuss science and its applications in our life. Next is a conversation with Katharina Hoff. Dr. Hoff is a world-renowned scientist with over 50 peer-reviewed scientific publications that received more than 4,000 citations in the fields of gene annotation, biotechnology, and bioinformatics. Her PhD was about gene prediction in metagenomic sequencing reads, and is now working as a junior group leader at the University of Greifswald as part of the Institute of Mathematics and Computer Science. She developed tools that scientists are using to find genes in living organisms. Also, she's a member of the Earth Biogenome Project Standards Committee on Genome Annotation, a project that is described as a moonshot for biology that aims to sequence, catalog, and characterize the genomes of all of Earth's eukaryotic biodiversity. Katharina is a genuine, friendly human being, a windsurfing instructor, and defines success as the ability to create something in science that someone else can use. This conversation is about finding genes, cells, and life. It's about science, diversity in academia, dealing with conspiracy theories and wrong information on social media and the internet. It's about motivation, failure, and success. I learned a lot in this episode, and I hope you will too. I am Ahmed Nazal, a medical doctor and neuroscientist in Heidelberg, Germany. This podcast is my own voluntary personal effort to bring science and its tools to the public at no cost. This episode is not supported by anyone. To support this podcast, please visit my Patreon page at patreon.com/amdlab as shown in the description and the pinned comment. Sharing this episode, commenting on it, and giving it a like will help the content reach a wider audience, and I thank you for that. This episode is in English, but I provide a transcription in Arabic and English by just clicking on the captions button on YouTube. Chapters and time segments are also available for you to use to jump to topics that are of interest to you. And now, without further ado, my conversation with Dr. Katharina Hoff. Welcome, Dr. Hoff. Katharina,
Thank
you for agreeing to talk to me today.
I'm very happy to talk to you, actually. I'm very excited about this podcast.
Yeah, same here. It was honestly a surprise to find out, like, while reading your CV, that you did the same, like the PhD part of the molecular biology program in Göttingen, which is the twin sister of the neuroscience program that I also finished. That was a pleasant surprise. So maybe you want to talk us through your career progress. Like, how did you choose to become in the field of finding genes, biotechnology, bioinformatics? They are all like difficult terms, and maybe you could tell us more about those fields too.
Yeah, sure. So when I was in school, I was interested in many things. Like in the last 3 years in Germany, you have to pick majors, and I had picked biology as one of them. The other ones were German and English, but really biology was the fascinating subject. But maybe it's surprising for you, I actually wanted to study arts after school. But to study arts, you have to pass entrance exams where they try to assess your talents in arts. And I totally failed that. So after a long battle of not getting into art school, there was not much time. And I applied for basically all kind of biology related study programs. And I ended up studying plant biotechnology at University of Hannover. I did not want to study biology, like pure biology, because numerous people had told me I would just end up unemployed, which I actually now believe would not have been true. But back then that was the advice, like study something that has biology in it, but not biology. So plant biotechnology it was. And actually it was great at University of Hannover. It was really a good faculty putting a lot of work into that plant biotechnology program back then. And it even allowed me to go abroad and like have this fully incorporated into my undergraduate degree. I went to Sweden. That was my longest stay abroad for more than a year. And I started working on plant lipid research. It's something related to plant biotechnology. I had in mind making new biofuels and saving the world, of course. But this was really nice there. But I knew that I did not want to return to Hannover after that, because Hannover did not have a lab that would do biofuel research in the way that I wanted to do that eventually. So during my time in Sweden, I had applied to many master's programs. Actually, one of them was in Heidelberg, but my absolute favorite was the molecular biology program in Göttingen. Mostly because I knew that Ivo Feusner was running a group there. And I thought I could fit in there with my interest in lipid research. So that was my reason why I really wanted to go there.
I'm not aware of him. Him or her, or now we'll get into the pronouns.
Him. Ivo Feusner is a him. Biochemistry professor. Yeah. And for practical reasons, actually, I had done my bachelor thesis in biostatistics because I wanted to do it from abroad. Like I wanted to do my thesis in Hannover, but sitting in Sweden. So doing something with a computer made this realistic. That's why I started going that way. And when I was in Göttingen, then the small bio program, it has lab rotations like the neuroscience program. That means you basically get to try different fields of research, different groups, 3 of them for a couple of weeks each. So I did one in Ivo Feusner's group, which was great, actually. I did one in NMR, up the hill at the Max Planck Institute for Biophysical Chemistry and Griesinger's Institute. And then I did one with Burkhard Morgenstern and Peter Meinecke in bioinformatics. And actually, that was the turning point. Like, I really liked the biochemistry stuff. I did. But when I was working with Peter Meinecke and Maike Tech and Burkhard Morgenstern, I was like, okay, maybe I want to do sequence analysis. They got me. So I decided to do my PhD in that group, and my thesis was on developing a novel method for finding genes in something called metagenomes. Now, you have a lot of terms here that sound complicated.
So many terms. I guess we're going to start with plant biotechnology. Like, because biotechnology by itself is, some people have like, it sounds scary for some people, like people are changing the genes, or so what is biotechnology for you first, and then maybe you could.
Yeah, so it is about changing the genes for me, actually.
Oh,
so let's maybe discuss what a gene is first. Maybe we should start there.
Maybe.
So not only the plants, but also the bacteria. Yeah, let's think about bacteria. They're like small and simple. They have something called a genome, and this genome basically encodes the building plan for a living being. And that can be a bacterium. It can also be a plant. Complexity varies a lot, actually. And these building plans, these genomes, they consist of parts that actually have the instructions for what a cell should be doing, and other parts that don't have these instructions. They may have regulatory roles, like how to do something, when to do something, but like they are not like the building blocks. And basically, these building blocks, they are called the genes, and some of them, most of them actually encode for proteins. So they are like protein coding genes. Now, protein, you know, probably from meat and egg, it has a lot of proteins. So all these proteins are made from genes in the genome. And plant biotechnology now is about improving plants, for example, agricultural or horticultural crop plants, but it could also be like other kind of plants. Like it's about improving them by changing that building plan. And you can do that in very invasive, scary ways, by literally going into the genome, cutting something out, putting something in. Or you can also do it like in less dangerous sounding ways by putting pressure on a plant and plant breeding so that it will start doing things you want it to do. And you can solve a lot of problems with plant biotechnology. For example, you can make a crop less prone to be attacked by certain insects. Yeah, so you get a better harvest. You can make plants adapt to salty soil environments, so you can grow them in places where you otherwise couldn't grow anything. People are scared of it, yes, because particularly in Germany, Greenpeace is always protesting the GMO plants, because you can, of course, also do things that might be dangerous. For example, if you take a plant that you use as an agricultural crop in Europe, and you insert, I don't know, a herbicide resistance gene in there, and this crop has wild relatives, it's pretty likely that this herbicide resistance thing will like cross out of the crop. That's one of the things people are worried about. Another thing is that they are worried about the gene products being toxic. That's in most cases actually not the case. Like plant biotechnologists try to actually not do that. But yeah, I've heard them.
I like that because here we're trying to debunk all the myths related to biotechnology. I guess like people are so, and that's where like the communication comes in. That is important from the scientist part, because people are afraid that the scientists don't know what they are doing. That's the thing, and it might be true to a certain degree that we're entering a field that we're still like learning about. But maybe you could tell me more about it because you're the master here about it. And then how much can we expect to, like, if you change like a plant to be more resistant to something, for example, and it ends up like successful, is there like a guarantee that this won't change other mechanisms in the plant? Like, do we put like control? Are there like do scientists have like control pathways that stops things from going wrong, for example?
Well, not always. The bad news is sometimes we don't know what we do, apparently. I must say, I don't actively practice plant biotechnology, and I got out of this field with my bachelor's degree. But usually it takes a lot of prior knowledge to achieve something you want to achieve. One example that probably many people know are the in Germany, we call them the Anti-Matsch-Tomate, like the tomato that will not go foul as quickly as other tomatoes. This was basically achieved by inserting a certain gene. You need the knowledge of which gene you want to play with. If you don't have that knowledge, it's pretty difficult. You can achieve things without knowing genes, like classical plant breeding since, I don't know, before Christ has achieved things without knowing about genes. But if you want to do it in a targeted way, you have to know a lot. And I would say today you do have ways to check on whether something went terribly wrong in your manipulation. Because in principle, we can sequence genomes now. So we can actually double check what happened. I'm not really sure that's always done, though. I don't know.
Yeah. But that's where your work comes in, like finding the genes, right? Like
Yes.
That's where now afterwards, like you moved into bioinformatics, but you said something about metagenes. So we have the genes, those are like the map kind of for building things in the cell.
Right? What I said was metagenome.
Exactly. And then there is like the epigenome, and then you mentioned something
we're like really like a lot of terms. So a metagenome is basically a collective genome of a microbial community. So you can go, I don't know, into a field, take a soil sample. The soil sample will be inhabited by a lot of bacteria and archaea, probably some fungi. Maybe you have a bit of plant material in there. And you can basically isolate all the genomic material at once, make a big mix and mess of everything and sequence that. So that's a metagenome. And I did my PhD on finding protein coding genes in the prokaryotic fraction of such metagenomic reads. Now you said we have an epigenome. That is completely different story.
Right.
Basically, in the genome, I said before, we have the genes, and then we have the parts that are, I call them intergenic region. Yeah, they are like the parts between the protein coding genes. And then that's not all. The genome, you can maybe imagine it like a string. You have a long string, and then it doesn't sit in a eukaryotic, like a human cell in this string-like shape. It's packed, it's neatly packed, it's spiraled, it's wrapped around proteins. And in addition, there are some parts of that genome that's neatly packed and wrapped up that are methylated. And when we talk about the epigenome, we mean which parts of the genome have this methylation tag, basically. And this methylation tag controls on how accessible the genome is. So that's the epigenome. You can sequence an epigenome, which means you identify the nucleotides in a genome that have these modifications.
Very interesting, very interesting. So I was reading like the papers, at least one paper. I tried to read one of your papers last week, and I really felt that you were going on a hunt, but on a microscopic level, or like trying to kind of automate finding genes. But how did we, like humans, start look for genes?
Yeah, that is a very interesting question. And I actually had to check on that myself. So basically, we have to go back to the time when people started sequencing nucleic acid material. So the genome is made from nucleic acids. It's called DNA. And even before that, people had been sequencing RNA. So now how does that play together? We have that genome, that DNA, just imagine that ribbon line again. And before this is somehow put into proteins, that information, there's some things called transcription. So the small parts of the genome that encode for a protein, they are transcribed into RNA, different type of nucleotides, a bit less stable on the long term. And then this is translated into the protein sequence. And the first thing that was sequenced actually was RNA. But then they started sequencing DNA. And this was like in 1972, I think. 1972 was the first sequenced gene, which was a bacteriophage coat protein gene. And then it's like took a little while, but people got faster and faster and more efficient in like sequencing and analyzing genomes. So in 1995, we had the first complete bacterial genome, which was the Hemainfluenza genome. It's a bacterium. And in between that time, from 1972 to 1995, all the time, people were like observing. So how does this work? How does this transcription step work? How does the translation work? And they discovered there's the codon usage. So 3 letters on the RNA are basically always translated into one amino acid, which is a building block of a protein. And the first methods for finding genes or identifying protein coding genes, they purely relied on these observations about the genetic code, basically. The earliest publication that I'm aware of about predicting genes is from 1982 from James Fickett from the Los Alamos National Laboratory. And his method was basically a decision classifier that would say, okay, this nucleotide is protein coding or it's not protein coding, don't know. And it was such a simple algorithm that you could perform it without a computer if you wanted to. He basically used the knowledge that they had back then about codon frequencies. And I think one major advance that happened down the line was made, at least to what I'm aware of, by a scientist called Mark Bordowski from Georgia Tech. And Mark Bordowski had the idea of using something called Markov chains. You can use Markov chains to basically predict the state of something. You can have a succession of days and you maybe want to label the weather is sunny and the weather is cloudy, the weather is cloudy, the weather is sunny, and so on. And you can like try to predict the succession of weather with Markov chains, hidden Markov models. And you can do the same on a genome sequence. And this is what he implemented. And he had a paper in the early 90s, like 1992, I think, with James McIninch together. And that tool was called GenMark by then. And this is, to my knowledge, like the most fundamental paper that we are building on today, because until today, we're using at least something similar to hidden Markov models for predicting genes. So it's all about statistical models. And I think they started doing it because there was this need from biologists to analyze the nucleotide data.
Okay. So like going back to like our first intuition that there is something that carries, like as humans, like first intuition that there's something in our in cells or in organisms, probably, like that carries the information. I think this is like a very interesting turning point in the way we think about everything around us, like about living organisms. I'm not aware of the point where, like, or when did we recognize as humans that there is something in us or in the living organisms that gets like, that determines who we are. Okay, this will end up like a plant, or this will be a mouse, and then that this material like goes down in lineage or for the species and is preserved somehow. So this like this insight, I'm not sure that I know when did this happen, like this kind of eureka point.
It happened in 1866, basically.
1866, okay.
Yes, so there was a monk called Gregor Mendel, and he performed plant breeding experiments with peas, like the green little vegetable that you can eat. And he basically had like inbred lines of, for example, round-shaped peas and wrinkly peas. I think he also played with yellow and green peas. And he was just like testing what happens if I cross them. And he described, at least to my knowledge for the first time, that there must be an inheritable trait. But this was buried, this knowledge. Like he wrote it up, it was out there, but nobody remembered for a long time until the end of the 1800s.
That's interesting, because Mendel is a monk. Yes. And it all started by a monk, like the biological. Okay, very interesting. Okay, and then we recognize that there's building, like some information at least can be coded, and then we figured that out, and then came like Watson and Crick in 1960s.
It was all in the 60s. It was a little bit earlier. Like I think a lot happened in the beginning of the 1900s as well, where like the concept that there must be something that carries the information was becoming more clear and clear.
So Francis and Crick and Watson like took the credit for like discoveries made by a female scientist, right?
Yeah, I think so too.
Yeah, interesting. Yeah, you see like how like science bits and pieces from everywhere, everyone is contributing to like where we're at right now. So you mentioned like, and then you could, at the beginnings, you could do it manually, like predict what gene is coding or like define the genes. So I'm not like it's not clear for me, why do we need to find the genes?
Okay, so the thing is that the proteins have so many important and interesting roles, and biologists usually want to know how living things work. Yeah, I mean, obviously we're not robots, but there's some building plan that controls how things go. Like, why do we fall sick? Why does our food grow in this way or in that way? And proteins are not the only important component, but they are such an obvious important component that biologists strive to know as much as possible about them. And in order to investigate a particular protein, you first must know about its existence. And today you can sequence proteins. That's a whole different story. But most of the knowledge we have about proteins is from genome analysis, because sequencing genomes, complete genomes, is so much cheaper and easier than sequencing complete proteomes, obviously. So we want to know where the genes are because we want to know which proteins are there. And that's why humans are trying to find genes to do basically further downstream biological research. So we're sitting pretty much upstream. And you were like earlier asking, why is it difficult, right? It's actually super complicated what our cells can do. That's why it's difficult. A human cell works completely differently compared to an algae cell or compared to a bacterial cell. And even to bacteria, let's think about E. coli. Some people have heard about E. coli. And then maybe let's think about something else that they might have heard about. Pseudomonas is also a more commonly known bacterial genus. Even these cells work differently. They use different signals for this transcription from the DNA to RNA and for the translation from the RNA to the protein level. And they also use different signals for controlling when is a gene actually transcribed. They use slightly different signals for controlling when translation is, for example, inhibited, because this can also happen. And this makes it pretty complicated to replicate what the cell can do. And what we do in gene prediction is basically try to use some statistical mathematical model or modern machine learning techniques in order to replicate what cells have been able to do all along.
I mean, that sounds very complicated, because I've up to my knowledge is protein prediction is called the grand challenge in biology because of its complexity. Like, so I guess like proteins are three-dimensional structures, and they're made of, like you said, basic building blocks called amino acids, and then once you put like different amino acids together, and how many amino acids? I guess we have 21 essential.
It depends a little bit. So there are 64 codons because they are triplets. 4 of them are stop codons. They don't encode for an amino acid, and it's not a fixed number. We have actually different variants of the genetic code.
See, so that's the complexity, and then they form in different 3D structures, and so it's a pretty complicated thing to kind of predict the way the protein will look like, but it seems like easier to look for the gene that is responsible for producing that that protein. So you wrote like in your paper Bracker
it's
or
called
Breaker?
Breaker, like breaking something.
Right. Breaker 1, unsupervised RNA sequential based genome annotation with gene mark ET and Augustus.
Yeah.
Man.
Should I maybe explain what that title is even saying?
I think so. We could start from there. And
let's take it apart. So the Breaker 1 is the tool name. Yeah, this is just like some name we came up with. That has a history on its own. I'm still contemplating on whether I want to discuss that.
Okay.
But what the tool is doing is using RNA-seq data. RNA-seq data is sequenced RNAs from the cell. So I mentioned before that people sequence genomes, and very, very early on, they were even sequencing RNA. And through the development of novel sequencing methods, nowadays, it's feasible to sequence a complete state of transcription of a cell at certain time points under certain conditions. And that is what we call RNA-seq. Yeah, so basically, RNA-seq data is evidence of transcription of genes. And you can use this evidence of transcription of genes and map it back to the genome. Like you can project, you say, this RNA read, like this piece of information in the genome belongs to a certain location. And now we have skipped over one important detail. While in bacteria, genes basically consist of what we call open reading frames. That means in bacteria, there is a start codon, and then comes a neat succession of triplets codons until there's a stop codon. In eukaryotes, like in humans or in yeast, this is a whole lot more complicated because in eukaryotic genomes, we have interruptions in the gene. So there's a start codon. Sometimes it's not even complete. Sometimes the interruption is in the start codon. But let's say there's a start codon. There are some codons. They may not even be complete. And then there's a break point. And then there is some region that is not translated into a protein. And then continues the neat chain of triplets. And there might be another like interruption. These interruptions, they are called introns. And when you try to project these transcriptome, these RNA-seq reads back to the genome, if you are lucky, they are spanning such an intron. So they provide information about these interruptions that are pretty complicated to reliably predict. So RNA-seq data has a huge value for finding genes because it gives us the intron positions, basically. And then there were two other tool names in the title. One of them was GeneMark ET. GeneMark ET is a tool developed at the group at Georgia Tech led by Mark Borodovsky. That's the same guy I mentioned before who basically, he left a huge impact in the field, even already in 1992 and earlier. Actually, I really love that we're collaboration partners. But what GeneMark ET is doing in the core, like many other gene prediction tools, it uses some kind of hidden Markov model in order to predict genes. And what all the other tools can't do, it can actually self-train the parameters of the statistical model if you give it a genome sequence and transcriptome data. Now, I said before, the whole gene finding problem is super complicated because every species basically has its own way of doing transcription and translation. Overall, they are similar, but they have those differences. And what GeneMark ET can do is basically adapt parameters to these differences fully automatically. Now, Augustus is yet another tool for gene prediction. Augustus was initially developed by Mario Stanke, who is bioinformatics professor in Greifswald. He's actually my boss. And by Stefan Waag in University of Göttingen. So Mario is also from Göttingen, but from the math program. And Augustus is also a hidden Markov model-based gene finder. And all these different hidden Markov model designs, they are like slightly differently. They are not identical to each other just because they sound so similar. And Augustus turned out to be very accurate compared to most other gene finding tools. But Augustus cannot do the self-training. So it's something we call a supervised method. So what Augustus needs is if you want to work with fly, it needs genes from the fly to adapt parameters in order to find genes in the fly genome. Sometimes that is a feasible scenario. For example, when he started working on fly genes, there were fly genes known, so that was not a problem. But today we're talking about sequencing thousands of genomes, and then it is completely unfeasible to have someone manually annotate hundreds of genes in order to train a gene finder. So the whole idea of BRCA1 was basically to combine the self-training GeneMark ET with the transcriptome data during training with the gene prediction tool Augustus, which can also use RNA-seq data, but in the prediction step, and have accurate gene predictions in a fully automated way. So this is a pipeline that biologists can basically start on their transcriptome and genome data and get a genome annotation.
That must be very important, right? But let me just go back into the steps. So we are talking right now about complicated statistical tools that verge at like the level of machine learning. Probably it is machine learning at the end of the day. So that is your like daily work, but for other people, probably like me, like I have some idea, but not in depth as you are. So you start with a gene. So like trying to understand what you said, like make a make a sense of it. So you usually you need like data sets, right? Like that's when you mentioned supervised learning, and then there must be unsupervised learning. And so supervised learning is, correct me if I'm wrong, it's like a way, so you have to tell like the function or the algorithm what the output is before, and there's like to train it on a human, and there's where like the word annotation comes in. So a human sits and says like, okay, this gene is that, this gene is that, or
Okay.
and
What we don't do in our group, but that is an important field as well, we don't say what genes do. We only say where they are. That's what our
okay,
tools do. Like saying what they do is something called functional genome annotation, where you say this gene is, I don't know, ribosomal rRNA, it's not a protein coding gene, or this gene is a peroxidase. Yeah, that's functional annotation, super important, but not what's happening in Breaker or any of our methods.
so for you, like the dataset labeling would be this gene is here, this
Yeah,
gene
it's
is
like this
there.
nucleotide is coding and this one is not. That's basically in the simplest way our labeling. It's a bit more complicated.
So
We
it's
do
like
not,
a
yeah,
0-1 kind of
kind of.
annotation.
Kind of, yes.
Or do you have multi-class labeling?
Well, we basically have labeling of start codons, like this nucleotide belongs to start codon. This belongs to a protein coding segment. This belongs to an intergenic region, belongs to an intron. This is a stop codon. So this is the kind of labeling that we have as input for supervised training.
And this is probably, so breaker one does not need that.
Yes, Breaker One can basically even like that's not in the paper that you read, but it can even run without the RNA-seq data. In theory, or also in practice, but I don't advise to do so, it can work with the genome sequence only. Because GeneMark ES, it's called ES, but it's in the same tool basically, can work on a genome sequence only, but it works much better if you have some kind of supporting evidence like RNA-seq data.
And who would use that? Who would benefit from breaker one? Like
basically all those little groups and individuals who are now sequencing genomes. So there's a bit of a difference. There are some huge institutions who annotate protein coding genes. One name you have probably heard is the National Institute of Health of the United States of America, the NIH. They have like, let's call it a annotation department or even maybe several groups. And also the European Institute for Biotechnology, the EBI. They also have their annotation groups. Then there's something called Ensemble. They also do genome annotation. They, in part, also benefit from this. I know that Ensemble people are now running our Breaker pipelines, not so much Breaker 1, more like Breaker 2. But mostly it's the individuals who sit there with their novel genome data and want to find the genes. So Breaker can or should be accessible to a large number of people. Apparently, it's still difficult for people to run, they keep telling me, but it should be easy.
Is it like written in a specific language?
Yes and no. Originally, it was written in a language called Perl. So Perl, to my knowledge, comes from the language processing, like natural language processing, programming languages. It can be used to also analyze like German language if you wanted to, or English language. And languages and DNA have a lot in common. There are a lot of similar methodologies that you need. Like, for example, you need pattern recognition. And this is something that Perl is strong in. However, the choice that we use Perl was rather historical because we had a Perl code base already. Now Breaker also has some Python components because we have pretty much completely switched to using Python in recent years. Python is not as specific to language processing, I would say, but it also brings everything you need. Python is much more universal. It's also the language for modern machine learning. You can do statistics with it. And the like under the hood, like the tools that are runtime consuming, they're written in C++. That's like
Okay,
Augustus is a C++ tool, for example.
I see. Yeah, so they are like toolboxes that you can use, like libraries that now. So you migrated to Python, and so like once you wrote Breaker 1 in 2016, and that was like when deep learning started to become kind of efficient and popular. Like in 2015, deep learning started to be a thing more than previous like support vector machines or other statistical tools that are not as deep as deep learning. So is it something that you guys are using now, like implementing in
Yes and no. Yes and no. So Breaker, the paper came out in 2016. It was a very lengthy review process, I must say. Not the longest we have seen. Now our new record is a year and a bit more, but
wow.
that back then was already long. The idea for Breaker was born at the Plant and Animal Genome Conference in San Diego, and I believe it was in 2013 already. And it had been around as a pipeline for a little bit when the paper appeared. So yes, that is in parallel to the rising of deep learning. And people in our group use deep learning. For example, one of our PhD students, Felix Becker, that is using convolutional neural networks in order to basically replace sequence alignment. So sequence alignment is basically when you have one or more sequences that you try to arrange in such a way that the matching letters are in the same columns. And he is using deep learning for that. Also, we are using machine learning for predicting whether a nucleotide is protein coding or not. There was a paper this year. I'm not on that. That's the work of Mario Stanke and Darwin Mertsch, actually, called CLAMSA. So yes, machine learning has its impact, its influence, but we are like not moving towards using machine learning only, like the modern machine learning, the deep learning only for genome annotation. There have been efforts. There are some manuscripts that you can find online, but they have not been like the big breakthrough yet. You never know what's going to come.
There were, I mean, that's pretty, yeah, that's a very good way of putting it, because sometimes if it's like adding deep learning probably without adding the accuracy or improving the output, you're just complicating things. And if you can do it with, I don't know, like a simpler, and I wouldn't call it simpler, but like less probably computationally costly efforts or methods, probably that's even better, I guess.
Yeah, actually, that brings me to a different thing, to explainable AI. So one of the problems that, as you just said, sometimes it's like machine learning with neural networks basically produces a black box. You can train basically a predictor to do something, maybe predict a gene. Yeah, you can do that. In my PhD thesis, we used a neural network in order to predict a protein coding gene in bacterial sequence chunks. But then you don't know why exactly is this black box predicting this gene here. And there is this new emerging field called explainable AI, where you can try to figure out why did it predict it this way. I know that Mario has, for teaching purposes, made some interesting experiments with that. But as I said, it's not something that has arrived at replacing hidden Markov model-like models in genome annotation yet.
Okay, excellent. So you are also a genome annotator part of the Committee for Genome Annotation in the Earth Biogenome Project.
Yeah.
That sounds like a very ambitious project.
Actually, it is. The Earth Biogenome Project is basically an initiative to sequence all eukaryotic life within the next decade. Disclaimer, it started a little while ago. And with all optimism that I have in my heart, I'm not sure it will happen within this decade, but yeah, something down that timeline, yes. So it's basically a worldwide initiative that's embracing sequencing efforts on different continents and different countries. And I'm not an annotator in there. I'm in an advisory committee. So the Earth Biogenome Project is trying to give advice to all these people who are doing all that practical work of sequencing, assembling, and annotating genomes on how to do it. For example, there are also committees on sample collection and processing, or on data analysis more generally than the annotation. Yeah, so I'm on that subcommittee where we try to basically come up with guidelines of what should be done at a minimum so that we say this genome has been annotated, and what could be done in addition, what do we recommend? So
for all life forms on Earth.
Yeah, basically.
That's...
Well, eukaryotic.
Eukaryotic. Yeah.
We haven't explained to anyone what's a eukaryote,
we
right?
have not, right?
Oh, God. So bacteria basically have their genome swimming around in that single cell that they are. And in eukaryotes, there's something called a nucleus. So there's like a double membrane layer wrapped around the genome inside the cell. So when I say all eukaryotes, I mean all the living beings that have a nucleus.
So what living beings don't have a nucleus?
Well, bacteria, archaea, and then there's this gray zone whether viruses are living beings or not. I tend to say not.
Well, they cause a lot of problem, like in the pandemic, the viruses.
Indeed.
So we need to take them serious. Okay. Yeah, that's interesting. So, like all eukaryotic cells, it's like the mitochondria in our cells used to be a bacteria, or...
Oh yeah, that's the so-called endosymbiont hypothesis. So the imagination that we have today of how eukaryotes acquired something called organelles is basically, yeah, that some larger prokaryotic cell somehow engulfed some smaller bacterium, and that smaller bacterium just lived on happily ever after in that cell. And that's how we think we got mitochondria. They actually even have their small own genome.
That's
it's
why
like a tiny bacterial genome inside of eukaryotic cells, and also the plastids, like the chloroplasts, for example. Similar hypothesis that small photosynthetic bacteria were basically taken up by larger cells and yeah, live on happily ever after.
Exactly, like it came to me because you said like the differences between the DNA, because the DNA in the mitochondria is they have like little DNA, right?
Yes.
And it's a bit different than the DNA in the bigger cell that the mitochondria is in it. And
yes, like... it's like a mini bacterial genome, basically. Same with the chloroplasts. It has lost a lot of functions. So both mitochondria and chloroplasts will not live happily ever after if you take them out of their host cell, because they're totally dependent now, because they lost parts of their genome. But they still have the most important things that are not encoded by the host.
That's crazy. If you think about it, like the level of complexity that we have to understand, we're trying to understand, it's like amazing. And thank you so much for clarifying it. I mean, pretty astonishing stuff. So let's kind of move a little bit away from those topics. And so I found you on Twitter, and it says that you are genome annotation geek.
Yeah, actually, that is funny because I'm not a Twitter person. I mean, I am on Twitter, yes, but I don't use Twitter much. Until very recently, I had maybe 10 or 20 followers, I think, because I only made a few tweets.
Okay.
I think only last week I landed my first viral tweet.
Oh, very good, very good.
And now Twitter might go down. Yeah, so I thought about using Twitter more for science because I have many colleagues who do that. Like I asked them, like, why do you do it? And they're like, well, we see all the papers we should read. And I'm like, yeah, but I see them on ResearchGate. Yeah, but Twitter is good sometimes too. So that's how I thought, okay, maybe I do have to use Twitter more as well. But that's funny. I actually thought you had found me through the MolBio program.
Yeah, no, actually, like through LinkedIn and then Twitter, and so that brings me like, do you think that it's important for scientists to have a social media presence, like these days?
Yes, and I have like a bad conscience that I have ignored that myself for a pretty long time. So, what I have always had and valued was a website that says like who I am, like what I do, what I'm interested in, because I usually check out scientists I'm collaborating with as well on their websites. If they don't have a website, that's a pretty disturbing thing. It does happen because not every institute gives a website to every PhD student and postdoc. We were very lucky in Göttingen that we always had that privilege. But yeah, social media helps like to go beyond this like kind of online stalking of other scientists. You basically get a chance to, yeah, basically listen along what people are doing, and that's a very valuable thing.
I agree. I learn a lot from like the accounts of researchers or scientists on social media. LinkedIn, I'm more active on LinkedIn. I'm trying to crack Twitter, but I don't know how to. I don't know, like it's not working for me. And but I see that some like posts like snippets of their research there, and they tell the people in easy ways, like understandable way, communicate their science kind of.
Yeah,
And
also like programming guidelines. I was actually showing my students in the classroom. I actually put Twitter on the projector this week, and I was like, look, if you have problems with Git, maybe you want to check this thread on Twitter because it gives really good advice on tackling your specific problem. But it's also like using Twitter takes so much time. Like you're like scrolling endlessly if you're following more than one person on through what's happening and what other people have liked and so on. It maybe takes a bit too much of resources to do that
maybe
for
we
a longer
could like
time.
tweet for Elon Musk now that we need a way to optimize finding the information that we're looking for on Twitter.
Well, a lot of people have moved away from Twitter already to something called Mastodon.
Oh, really? I never heard of it.
Oh, yeah. It sounds like a, I don't know, guts disease, but it's actually a software tool similar to Twitter, but totally decentralized. So Mastodon basically operates via decentralized servers that are funded by different communities.
That's cool.
And so far, Mastodon is so easy and quiet for me because not everyone has moved yet. Like a lot of the people whom I appreciate are there. Like, for example, I really like to read about the things happening in the group of Rob Waterhouse. He's also involved in the Earth Biogenome Project. He's on Mastodon. So now I see all his tweets or Mastodons, whatever you call them, very prominently because he's one of the few people who say something. But yeah, I don't think there's a point in talking to Elon Musk at the moment. No.
I see. He's busy like firing people and hopefully hiring others. But yeah, that's interesting. But the problem with social media and is like fake news get more attention and people consume it faster, and like it spreads, like false information. That's like a huge problem. It spreads faster than true, well, evidence-based facts. And I wonder, like, why does it have to do with the way scientists communicate with the public, or is it just like the nature of us human beings, like wanting like conspiracy theories? They are easy to understand, and it gives like a simple solution for a huge problem, and then you're like you don't need to put so much energy trying to understand what's going on. like in comparison to scientists who would dig deep. And I don't know, like it's really a big problem in social media. Like, and every platform has this problem right now.
Yes, actually, I had the most shocking encounter. Like for me, it was really shaking me in my core. I was sitting in the practice of my physiotherapist. So I sometimes have back problems. So I see someone who practices osteopathy. And I was sitting in the waiting room, and then eventually he called me in, and he was very upset. And I was like, oh, okay. And then he started telling me why he was upset. It was because of another patient who believes that we are all controlled by reptoids. Like seriously believes that. Like I never thought it's actually possible that a person believes there are reptoids among us who look like humans and who control humans and have been doing that for the past I don't know how many thousand years. And then I started thinking like how is it possible that someone believes that? And yeah, you're probably right. People look for simple explanations, but reptoids are not a simple explanation.
Probably like like that's the bits and pieces from here and there that you hear, and then you kind of like the brain tries to make a story that is easy for you to understand. So maybe he heard that some scientist says, oh, we have the reptilian part in the brain, that is, or he said, okay, and then he heard someone else that's talking about aliens and reptiles that are intelligent, and then made the link, and it made sense for him. That's the thing. Like, you don't know what makes sense for people, and is it correct to kind of control the information, or really have like control over the conversations that people are having, or just
I think it's important to like stop crap, like really incorrect, totally wrong information. I think it's an invasion of privacy if someone went into like a private one-on-one conversation I have with a friend, like imagine I believe in reptoids and I tell my friend, oh, I think today I saw Angela Merkel. I think she's a reptoid. Yeah, that would be wrong if WhatsApp would intercept, like, no, you cannot tell your friend you think Angela Merkel is a reptoid, which I don't. Yeah. But if it's like semi-publicly in a larger bubble, I think we absolutely need to get this under control. I mean, one thing, like that's why I was so excited to talk to you, is that we need better science communication with the public. Honestly, I do have some doubts that the people who believe that Earth is a flat disk, despite every knowledge we have today, are going to listen to a science podcast. I was thinking that a larger effort needs to be put into schools to make it very clear to kids of every age until they get out of the school system that, these things, they are like not true and other things are true. But then on the other hand, it's a problem. We have religion around, right? Religion is pretty much for me similar to the reptoid theory, just that it has been around for a much longer time. And then there are teachers who teach creation theory, like they teach kids that there is basically like an equally justified model of how humans became to exist, and that is that God just created you on day so and so. And as long as we don't have that under control, I don't see a chance of getting the other things under control.
Complicated topic. And the thing is about people who are into those, they defend those things fiercely, because there's an issue with, I guess, the mechanism to filter information and the understanding of reality, and then brings you to what reality is, and like the common reality and the individual reality, and what we agree on to be correct reality, truth, facts. It's a difficult topic, and I agree with you. Like, so there should be some kind of form of control, but not controlling what the people say, but like maybe controlling what gets disseminated, because we know that social media companies are able to control which content gets more likes or gets more shares, and they could monitor the quality, who is producing that information, and in specific topics kind of like make sure that authorities are, or like trained individuals, not authorities, trained individuals that know what they're talking about in the specific field. They are kind of the ones who the algorithm would suggest to people to watch. Because otherwise, because the algorithm goes the other way around. For some, it just feeds the people what they want, and they are, if they have already skewed version of the reality, then the algorithm will just go with the flow, and there is so much content that probably more entertaining than just like a scientist or a science podcast, and people would consume that more, and then the algorithm would keep on, oh, I think, yeah, maybe that is some good content, and then would promote it and just like keep on showing it to other people. I think it's a big topic.
I think one thing that could be made more clear even to kids in school is that we are not on a frozen solid knowledge level. Like all these conspiracy theories, they have some ground truth. They like to cite some paper from someone at some point in time and say, but he said the masks decrease the oxygen level in the blood. It's a scientific publication, something like that. It's important that everyone understands, yeah, he might have said that, he might have had scientific evidence, but it's a flow. Like after this came another, I don't know, 100 papers who said something else, and they found a flaw in the methodology. And even those 200 papers might be wrong. Like next week, we might have someone else who finds out something else. Like this is difficult for a lot of people to understand, that science is not a frozen state.
It is difficult, and like Alzheimer, for example, I don't know if you heard about this, but there's a mainstream theory about Alzheimer that it is caused by a certain accumulation of protein in the brain, and it was found that figures and all the data in the most important paper were fabricated, and that led to 20 years of research and people wasting their life, their taxpayers' money, and down the drain, basically. And our understanding of the whole disease and is still did not improve. So that's the thing, ethics in science and is very important, and building trust between the public and the scientists is also important. And the only way, I guess, is for the scientists to go just like what you're doing, and tell people what they are doing and what they are working on, and have a conversation, basically, even no matter what the conversation is, but be out there, tell people, okay, this is wrong, and or this is, or yeah, that and we did this because of that reason, or we changed our opinion because of that. Just like Karl Lauterbach changed his opinion about cannabis legalization, for example. So, yeah. Well, such a difficult topic to discuss, but thank you for going there. And so, in science also, I wanted to ask you, how was it for you? How was your experience as a female in or a woman in the scientific field, in a technical field? Do you face any obstacles that you think a colleague, a male colleague, did not face? Or
so I have been very, very lucky. That is one thing. I have always worked in groups that had a pretty good gender balance, I would say. I have never worked in a team that would consist of men and me. For a very short time, my current group was dominated by men, but also that has changed again. It depends on when you hire people. But so I have never felt like I don't know, someone is giving me a difficult time because I'm a woman. What I find very challenging is the academic system, like the way the system works. That's not like a person who comes to you and says, you don't get a promotion because you're a woman. It's more like, if you want to rise in the academic system, you have to be very flexible. You have to be willing to move to different locations. And that kind of is a problem if you have a partner who's older than you are. And that happens for a lot of women and for me too. Not for everyone, but my husband is 9 years older than I am. So his career started earlier than mine. So he got the really cool job in 2010. He got his professor position at University of Greifswald. I was basically ready to move on to a professor position so long ago. In like 2019, I was offered a professor position at University of Wageningen. And I didn't move there because I have a child, I have a husband, they're in Greifswald. Not so easy to take them along. So this creates serious problems, and I think it happens more often to women than to men, because us women, we have this weird preference for the older men somehow. But I would not say that I was discriminated for my gender. That did not happen, but basically, particularly having a kid and being tied to a geographic location makes it challenging.
So it's like more of a biological disadvantage then.
I'm not sure whether this is a biological thing, how you choose your partner, or whether it's also something, I don't know, passed on through our society. I'm really not sure about that. And it's not like there are no solutions. There are many couples in academia, and some of them have like their little deal, like, okay, I get my professor position now. If you later get the chance, I'll pack and drop my things and we move to yours. You can have these agreements, and maybe I should have made such an arrangement a long, long time ago. I didn't. Actually, I thought for a while that I would be very happy to be on the postdoc level forever. Like I really thought that when I moved along to Greifswald. It took me quite some time to realize that I really appreciate the freedom of running my own little team and conducting my research more independently.
Yeah, that's interesting, because like what I'm noticing now is like there's an exodus out of academia to fields like in the industry or other fields. Or is that your perception
You
too?
mean in Germany, or for women, or in
general?
Russia?
In general,
Okay.
no. In general, like people are leaving academia because and postdoc positions are like not unbefristet, and like they're not they're limited. There's problem with funding. I don't know. What do you see like the
Yes.
pros and cons of staying in academia?
So first things first, I'm very lucky. I got a permanent contract very early on in 2013. Those positions are super rare, so I'm blessed to have it. I would have left academia for certain without the permanent contract. I would never have continued this. The problem is that science has to be competitive in order to move forward. So science in Germany is mostly, I'm not talking about industrial science and research, but about the publicly funded science and research is funded by the public. Like a taxpayer is funding the universities and also the research institutions mostly. And if we want to invest tax money, we want to invest it smartly. Like we want to get the biggest outcome of this. And also we don't want to put in indefinite amounts of money because we don't have indefinite amounts of funding. And that means you're trying to select the best in conducting science, the best in teaching, because you want to like teach the next generation to keep the system running. And this has led to a couple of arrangements in the German academic system that are like super challenging now for young scientists. One thing that happened was that a lot of permanent positions were eventually made temporary. I'm not sure about the exact time point, but I think that was changed like in the 80s, 90s, in order to make it possible that our system remains competitive. Yeah, that was the whole idea. But also like the laws for work contracts. Basically, usually you cannot just hire someone for a short period of time and then say, I don't need you anymore after 3 years. Like that would be a typical PhD time span. So we made our own law, our own exceptions in order to be able to hire scientific staff on temporary contracts. It's called the Wissenschaftszeitvertragsgesetz. Which basically gives scientists a limited number of years, typically 6 years before PhD, 6 years after PhD, when they can be on time-limited contracts for their own qualification. First qualification level is obtain PhD. Second qualification level is usually obtain habilitation or equivalent. So we have this law, and this law creates a lot of problems for a lot of people. And I understand everyone who doesn't want to do it. Oh,
no,
habilitation. Yeah,
is it like assistant professor kind of?
No, so that's pretty old school concept. A long time ago, a PhD system was already established. We called it doctor, not PhD in Germany. It was assumed that in order to be able to teach other students, you should have written a second thesis and you should have proven that you're capable of teaching. So you can do basically a second PhD. A second doctoral degree. Usually you're supposed to do that with a lot less guidance than the first PhD. And actually, I believe that's why I said it's very old-fashioned, that habilitation is a dying concept, because in the US, for example, you don't have this. They have assistant professors who become assistant professors at a quite young age after the PhD, and then you can rise in ranks to associate and so on. In Germany, we actually tried to reform the system as well. We created the junior professors, which are like assistant professor positions. And today, when you apply for a full professor position, you don't have to have the habilitation. If you, for example, did a junior professor job before with positive evaluations, that's pretty much the same as a habilitation. And even if you didn't do that at all, like most of the bioinformatics professors, they have never done the habilitation. I did it, but I only did it because I don't have a professor position and I want to be able to supervise my own PhD student.
I see. And do you have to apply for grants or something in this habilitation?
I think it doesn't hurt, but it's probably field specific. So in the natural sciences, we are used to applying for grants. I'm not sure whether you have to show that you were successful in getting grants, for example, in law. I really don't know.
I see. Okay. So I have like a question in general about science. So as a scientist, you're used to deal with uncertainty, like trying to find things that no one found before, usually. So how do you deal with this feeling of uncertainty that comes with trying to figure things out?
I never saw that as a problem, actually.
Okay.
It's I don't know whether that's a matter of personality. I'm not sure, but I never had this urge to believe in something. I was baptized as a child. Like my parents didn't ask me; they just baptized me. And I also went through this kind of church school as a teenager. I never identified with that. Like that was like believing in things just for believing in them is never something that worked for me. And then if you don't want to believe in something, you just have to establish for yourself that nothing is for certain. Yes. But this, like in science, nothing is for certain. In terms of how you want to arrange your life, I actually value very much to have permanent work contract. As I said before, I would have left the field if it wouldn't have been for a permanent work contract. I would not have had the patience to hang in there for, I don't know, until the 12 years were up and always say, oh, I hope in the next 6 months I get another extension of 6 months.
Yeah, that's true. And so the unknown never scared you?
Well, my husband says I'm generally easily scared. So I wouldn't say that. For example, we went snorkeling in Thailand in the beautiful blue ocean. and we didn't go snorkeling at a coral reef, but we took some canoes and were paddling between islands, and you can't see the ground when you're snorkeling there. It's in the deep water. This scares me. This freaks me out. Like I was like, I don't want to be in the water. I want to be in my canoe. Let me back up. So I am scared by the unknown, if it's visual for sure. But I'm not worried about whether a finding I make today may be overthrown by tomorrow. That will probably happen.
Yeah, that's part of the scientific process. So you had no problem dealing with failure then of an experiment?
Oh, I have a lot of problems with failure. That's a different story.
Okay.
But there are two reasons for failures. One reason for failure is because your hypothesis is wrong. This is something you have to expect, in my opinion. It can happen a lot. The other reason for failure is that you mess up as a human being because, I don't know, you're clumsy and pipetting things in the lab, or you're a bad programmer, which would more apply to me. And then this feels really, really bad if you fail. If you fail because of your own shortcomings. And well, that hit me pretty badly, actually. So I came from molecular biology into bioinformatics, and during my PhD, I was, let's say, very well protected by my surroundings. Like I was a really bad programmer. I was able to do what I wanted to do, but it was not pretty at all and not reusable code either. But people around me compensated for that. I was so lucky. And then I moved on to my first postdoc phase, which was with Tim Weisbart in Göttingen, but that was so brief. Then I moved on to University of Greifswald with my first own grant. And then I realized that I really have to learn so much because I can't compete. And then this was really exhausting and really tiring for me to catch up and very frustrating as well. So my way to cope with it was basically to improve my skills, like always try to improve my skills. And it's maybe not a healthy way to live. I'm pretty ambitious if I want to get something done, and I will sit at my computer long hours. I have a little daughter now. She's 6 years old. I'm the one who doesn't come home on time. My husband always does. I'm the one who continues to sit in the office to finish something because I was too slow in the morning.
I see. Okay. So you deal with it by pushing forward, kind of. I see. And so what keeps you motivated?
Yeah, it's a good question. Sometimes I wonder whether my motivation is hidden in the garage. No, I'm kidding. I am motivated by success. When you're a kid, you draw something, you go to your parents or your teacher, like, look, I was drawing this house for you. And they're like, oh, this is so pretty. And as a child, you feel so rewarded. They have to keep your drawing and put it on their wall. It's a little bit different, but in the field where I work, it works similarly. If I program something, a lot of people are going to run it usually, and they give feedback. I mean, in science, we have citations, like how many people basically quote using your method in their paper. This is almost like the, oh, you draw this in such a pretty way, rewarding bonus thing. You also get a lot of not so nice feedback when things don't work out. But basically, my motivation comes from knowing that I can expect kind of a reward in some way.
That's pretty much the way it is, right? In our brains. Like,
yes.
We work something because we expect a reward to it. Very good.
Exactly.
Okay, you brought the term success, and I tend to ask, like, what does success mean to you?
Well, success is for me that I create something in science that someone else can use and will be at least a little bit happy with. Or it can also be that I teach students and afterwards they actually know what I try to teach them. They don't always do, but most of them try. And then they say, oh, now I understood how, I don't know, the correlation analysis works. And that is also a success. It can also be that I teach my daughter something and she's like, oh, okay, that's why, I don't know, the plant is green. We had that discussion. Yeah, that's the small things, basically.
Yeah, it is always the small things. I ask this question every time I meet someone lately in this podcast, and it's I'm like maybe by the 100 podcast, I will sum it up in a little book called like what success means for you. I don't know.
And then you can have a psychologist profile us and identify, oh, Dr. Hoff has the narcissistic tendencies. I probably do. But I know that if you ask colleagues of mine, the answer will be very different.
Yeah. Okay. So, I mean, I have just like one last question left, and it is, since you're working with genes, localizing them, I think maybe could tell us how long do you think humans still have on Earth?
I have literally no idea when we're going to blow ourselves up. Do you know this YouTube video, The End of the World? If you don't know it, put that line into YouTube. Maybe you want to put the soundtrack into your podcast.
Okay.
It's basically about how we're going to end life with nukes.
Is it like a little cartoon or?
Yeah.
I think I saw it.
So the thing is, like, I have no idea when we're going to destroy our planet. There are a lot of, I call them the young people. I feel so old right now with my gray hair, who are like trying to make things better, who are like really fighting global warming and all that stuff. And at the same time, we have crazy politicians who have like a power button to to launch a really bad rocket. So I have no idea. I hope we will have another thousands of years on this planet. I seriously hope that.
Do you think we need to be extra like multiplanetary civilization, or should we take care of the home that we know?
We should be sustainable with our resources. We should figure that out. But it looks like we will fail. Like we're on a good way to melt the Arctic poles and to have a big flooding and bad wars.
Yeah, it's like the data, I guess, seems just this way, but we have to believe like. But made us make it till here, I hope will keep us a little bit longer as a civilization. I hope.
We have survived a lot already. Like,
I think so, right?
If you look at how, for example, Europe looked like in the Ice Age, in the last Ice Age, and humans prevailed. So my personal future hypothesis is that we will have very wet feet in Greifswald because we are not very high here. So maybe people will stop living here eventually, but we also have high mountains.
Move back to Göttingen, the center of is it the center of Deutschland?
It's pretty central, and I really miss Göttingen. I do.
See, like we're gonna wait for that moment, and we all like collects back in the center of Germany.
It's like this PhD program there, and also the neuroscience program that's so closely related, has something really unique in making people connect, like besides the science. Like you have all these scientific PhD students who do their amazing projects, but at the same time, we always had all these parties and these meetings. And I'm in touch closely with a couple of people from my year and from other years all the time. Like that's I really beautiful. Value that friendship.
Yeah, it was very international. It was definitely like I feel like I was lucky to be part of that program. I remember like telling you know him like. Probably Horner, like, and first day I remember. I think I got lucky to get in here, and he said, "Yeah, I think you did."
Like, I felt so lucky. Like I got admitted on the waiting list.
Me too.
Yeah, and I had I had really been hoping for this, and I had dropped other offers for this. Like Heidelberg admitted me to the master's program in biotechnology, not in MolBio.
Hmm.
And I had not taken that offer, and it was like late September, and everyone was asking, Katharina, what are you going to do? I was like, I really want to go to Göttingen. I didn't get in
So
yet.
yeah. So you were into the like you knew the people who were in the program and you were interested to work with a specific that's great.
Yes,
That's great.
yes. And it worked out in the end pretty last minute. I missed orientation basically.
Mm, wow.
So I felt so lucky that it worked out. I honestly have no idea what I would have done because I had pretty much dropped all the other opportunities already.
I mean, Göttingen is beautiful. I live right now in Heidelberg, and for student life, it had that like specific atmosphere. Well, it's beautiful. Like it's down to earth, kind of real, and it's a bit different from Heidelberg. Like Heidelberg is pushy, kind of like I don't know. That's like at least my impression.
Oh
Um,
yeah. It's my
yes.
impression too from the outside. It's actually why I didn't want to go to the biotech program there. Now I can
So
say that.
yeah. So no, Göttingen is beautiful. It's a little bit colder than Heidelberg, but
Yeah, it is.
yeah.
I don't know. Now I'm in Greifswald, so Greifswald also has a very nice student life atmosphere,
Hmm.
I must say. Basically, the city is almost like dominated by students during the semesters. It gets pretty quiet during the breaks when people like go home to be with their families or
For
travel
sure.
or something. But what I appreciated a lot about Göttingen was that they always seem to have enough funding for the important things.
Hmm.
Um,
Yeah.
and that makes it very different. Like now in Greifswald, all my colleagues in administration, they are working super hard and they are super kind and I appreciate all of them. But it always feels like we're behind in terms of resources, financial resources, just
Hmm.
because we're in a different region.
That's true. That's true. Like Matthew's effect. I
Yeah.
don't know. The so yeah. I mean Göttingen and Greifswald. Very interesting stuff. I lived in different like university cities are for me are the best in Germany. I lived in other cities, bigger ones like Bremen and Berlin. But I decided to go back to Heidelberg because it has more sun, like on average, than Göttingen.
We
Uh,
have even more sun than Heidelberg.
really?
Yeah.
Oh man, that's very good. And you have like a beach and like it's a coast, like at the
Yeah,
coast,
actually, the first thing I did when we started working
surfing.
here, was surfing. Like I enrolled
Awesome,
myself in a windsurfing class. I even became a windsurfing instructor. Um,
man. Respect.
like without a license though, but the Hochschulsport, university sports didn't mind. But I had to stop doing that when I got my kid
Mm,
because it doesn't work with the additional hours.
right. Well, maybe now you could, once she's a little bit older, you could teach her, and then she becomes a surfer too. So, I mean, big cities are in Germany like they have a different atmosphere than university cities are really special. You have everything that you need as a student, and it's everything in walking distance. And people understand that even if you're international, that they have a little bit more understanding to you than I felt Göttingen like I had my connections like with Germans in Göttingen like more than other because I play music and I was lucky to kind of find German musicians to play with and so they even have an understanding and they were like yeah this guy does not know anything about here so let's be nice to him so that was nice I guess so what's next for the research that you are doing and
Oh yeah, what's next for me? So I did my PhD on predicting genes in bacterial and archaeal chunks and metagenomes. And that is a difficult problem because I said before that all the cells work differently, have their own signal machinery, and you cannot train the gene finders on very short chunks of genomic material. And there are some novel sequencing technologies, for example, a company called Pacific Biosciences, who develop protocols to make very long reads. There's something called the PacBio Hi-Fi reads. They're very long reads with a very high accuracy. So they're not having many mistakes in the sequence. And this makes it feasible to actually also predict genes in the eukaryotic reads. So a project I'm working on with my new PhD student, Natalia Nenasheva, is to make methods for predicting genes in eukaryotic metagenomic sequences. That's one thing that's next. There are many, many more, actually. I have a lot of loose ends that I want to catch up on, but my main thing continues to be making fully automated methods that achieve a high accuracy for genome annotation. That's still my thing.
Great. Okay, I wish you great luck in
Thank
this
you so
endeavor.
much.
Dr. Katharina Hoff, thank you so much for taking the time to meet with me. I learned a lot. I think we tried to cover so many things, from biotechnology to bioinformatics to your experience and to genes to DNA. It's too much to understand, too much to cover. And I'm very thankful for your patience and trying to communicate that to the audience.
Thank you very much. It was an honor to talk to you.